Thank you, Rhim, and thanks to RainDance for inviting me to come and present some of our data, and actually, in addition to our data, kind of present the concepts and some of the challenges that we face associated with implementing new technology. And I think this is the beginning of a really interesting story with respect to all of the challenges that we face in not just analyzing samples, but actually getting samples, making them last, and getting the most utility out of samples. So what I'm going to talk about for the next short while is, what we've done to employ the RainDance raindrop technology for both targeted sequencing as well as digital PCR. In this respect for some of our molecular oncology work, but hope to describe some of the more broad applications that we're applying. So just by way of a brief introduction, RUCDR Infinite Biologics is a biorepository program, a full-service biorepository program that enables clinical trial management right now for 5 NIH institutes, and we enable sharing programs not just for data, but also for tissue, for nucleic acid, and for study results that facilitate currently about $1.5 billion worth of NIH-funded research. We do this same kind of work for research advocacy groups as well as a number of private sector clients, and our goal really is to speed discovery by looking at the genetic contribution for complex diseases by sharing all of these samples and this data. We've grown over the years. We have a decent-sized staff and currently have close to 20 million samples under management. We also distribute these samples, which is an important point to point out, for people that are doing analysis in their labs. And this is where identifying and implementing technologies that use the least amount of sample really becomes critically relevant. So one of the things I want to point out from our perspective is the difference between storing your sample, what we call just biological storage, versus true biobanking. And there is a difference. Bank samples proactively acquired for future testing and analysis. And when it comes to clinical trials and in the past, samples were collected usually with one purpose in mind, to measure one analyte, and then were destroyed or really not thought of again. But now people are working very hard to repurpose, expand their IRBs, expand their goals to collect samples so that we can go back to them again and again to do different analyses for a number of different molecular compartments. So a bank sample is often sent to multiple different recipients. So in the case of a lot of our controls, we will send them to hundreds of labs for different types of analyses. So to this end, a bank sample should never be depleted. So we really do consider every sample a limiting sample. Even in the case of whole blood DNA, hundreds of micrograms at some day or at some point will become limiting. When we talk about our tissue samples, they start out being limiting. So when we think about implementing technology, we look at the 5 major functions of a program like ours, which goes from sample acquisition and logistics of actually sample transport, the processing of that sample, whether it's the fractionation of sample or the extraction of nucleic acid, the storage of that sample, the distribution to the labs doing analysis, or inline analysis. And this part is really where a lot of biorepositories are growing, in that we are getting to the point where more information is needed for each sample for those deeper analyses to be done. And when we start to think about what the importance is for these samples and the analysis that we do, there are a number of what we call the functional essentials for maximizing these biological resources. So the first is really thinking about the maximal use of primary samples. There are still undefined applications for downstream analyses. So if we look now, at ThunderBolts, a year ago, that didn't exist, right? So what are the qualifications for these kinds of assays or technologies? And this leads to, obviously, the increased use for these samples. So we work on efficient processing. We maximize extraction technologies to improve yield and quality the best we can. But at the end of the day, because of all those other 4 things that I showed you we do, sometimes there's not a lot of control. And I challenge anyone who's ever worked with anybody else's samples to say, well, you know what? We understand this sample's low quality. We don't have enough. We'll just skip that one and not run it. That's the one they really want the data from, right? So, even with the most qualified efforts towards efficient processing, we still have to look at technologies that are most robust, developing appropriate storage formats, so we can utilize these samples effectively without jeopardizing them just with the whole in and out of a biobank process. Creating renewable resources, looking at nucleic acid amplification schemes and cell line establishments. We create cryopreserved lymphocytes from just about all blood samples that come in. We did that a long time ago so that if we ever ran out of DNA, we could make more, like the Doritos commercial. You know, just keep eating, we'll make more. And interestingly enough, in the last 2 years, we've actually developed now an approach to creating induced pluripotent stem cells from CPLs. So this insurance policy now helps us create resources that otherwise didn't exist, you know, previously. So we think about these things in advance. And then also developing the appropriate distribution guidelines. So anybody that requests a sample from a biorepository, make no mistake, is making their own little biorepository. You know, I need a little more sample in case it fails, if I want to rerun it. Even though I signed an MTA, I told my friend they can have some. You know, all of these things are really being circumvented by generating more data on these primary samples using things like ThunderBolts so that we can use those samples for deeper analyses in the future. So this brings us to the RainDance Droplet technology. This is a cartoon that basically demonstrates from a complex target the ability to put single molecules inside these little droplets. The relevant applications, once these droplets are created, really are open to your imagination because we can run reactions in those individual droplets. What I'm going to show you is data from first next-gen sequencing through ThunderBolts. We actually use the same technology, the same source technology for generating droplets for digital PCR and get a readout, which I'll show you the importance of that to us, and then certainly the ability to use the limiting or unmeasurable amounts of sample that are present. So on the digital PCR side for target validation, it's a similar approach. The sample or the complex sample, which has your target and limiting amount in the context of a lot of background, is divided up into individual droplets with a single molecule ultimately in each droplet. We then do standard PCR amplification using chemistries like TACMan to then be able to count real-time the number of hits in total, which represent a hit from each droplet. This kind of digital counting is an endpoint assay that allows us not just to validate things in our clinical lab and for our research applications, but also is being used independently for us to qualify and look at the quality of sample. And the reason that this is important is when I show you in a few slides our DNA workflow and our analysis and QC workflow, up until recently, we used technologies that really were commensurate with what people were using for discovery, i.e. just regular genotyping. So I would say out of the close to 1 million samples we distribute a year, 90% of those or greater were used for standard genotyping, whether they were individual targets, microarrays, or what have you. But now the shift is really towards analyses with a higher resolution. And people are sequencing very deep, very specifically, and we're starting to wonder whether or not our quality control metrics will support the sample quality for those analyses. And what we're always concerned about are sample collection errors, contamination. So if we look by TACMan just for QC, knowing that this is the approach people are going to use downstream, that's fine. But now we have to kind of graduate to more sensitive single molecule detection to make sure the quality of our samples are commensurate with what people are going to use them to discover. And then the last workflow slide here shows the ThunderBolts targeted enrichment. Again, I think you can see the theme here. You start with a complex sample, and you end up with lots and lots of little droplets that have individual molecules. Again, the raindrop source is used, and right out of this ThunderBolts protocol is a sequence-ready product. So again, in a program like ours, when we're dealing with thousands of samples on a daily basis, having a technology that allows us to go directly into analysis, even 2 hours saved on a protocol, adds up to an immense amount of time for implementing on a large scale. So the hotspots or the important genes that we're looking at in this panel were really well selected. There are 50 genes covered by 230 amplicons. You can see the targets up here. I'm going to show you our validation, which was on colorectal samples. We compared fresh frozen along with FFPE samples to look at the efficiency and the accuracy of the panel. So there are a few markers that I'll focus on here just in the interest of time. The important thing to point out is I really think the RainDance team did a great job in identifying what targets there are by looking at what's out there in the marketplace, right? So it's, you know, it's not an easy thing to come up, obviously, with novel targets necessarily, but we can learn from everyone that's come before us. And I think this is a good example of what's been done there in that, you know, the entire AmpliSeq panel, as well as a lot of the content from Illumina TrueSeq has been incorporated and really fine-tuned to get us to the resolution that I'm going to be able to show you. And this is just, again, another RainDance slide that just shows the simple workflow from sample to sequence. We start out with, you know, very limiting amount of sample. We extract the sample, and again, it's really agnostic to the type of chemistry that you're using for extraction, but I'm going to show you how we account for that. It goes into the raindrop, runs the ThunderBolts protocol, and then goes directly into sequencing and analysis. So when we think about, on an enterprise level, how we implement technology, there are a number of considerations that we have to take into account. And there are so many new technologies. As much as we would love to play all day long with all the new toys, there's just not enough time or resources to do that. So we have to very strategically choose what we're going to evaluate. And given our experience with the raindrop for digital PCR, you know, ThunderBolts was a natural progression for us. So first we look at specificity. So I commented on the content selection, and certainly accuracy is critical here for the level of sensitivity that we're looking for, which is the second consideration. How can we really maximize the amount of starting material? And with that limiting amount of starting material, how good is our detection, right? So we treat every sample like it's limiting. We don't ever want to have to disqualify a sample, so what can we come to expect for detection? Applicability, does it meet our programmatic goals? You know, are the panels well thought out? Is there ability to customize in the future? We were able to, you know, check the box on all of these things. Flexibility is key, so we certainly want to maximize the utility of our infrastructure. So being able to generate our variant data and validate it on the same instrument is a huge advantage. And certainly in the CLIA environment, if you think about it, it's also an advantage. And actually, the opinion that we've gotten from our QA group and our CLIA lab director is it's really the assay and not the box or you know, the underlying technology that is required for validation. So TACMAN chemistry versus sequencing is a viable validation, and the fact that we can use the same instrument makes our validation and our QA process a whole lot more straightforward. Efficiency, workflow, implementation, certainly, like I said, the fewer the steps, the more robust the assay makes it very efficient for us. Scalability, you know, does the technology meet our throughput goals? And then ultimately cost. You know, you could put cost first or last, but it factors in quite significantly when you start to talk about sample numbers on a considerable level. And that's really what enables global implementation. I could tell you over the years there are a lot of great technologies that we validated, that we love, that quite honestly we didn't put into play because the budget just didn't support the kind of data that we get out of it. And we would do it in a heartbeat, but the technology could never achieve that level. So let me tell you about our study. The study that we did was using some colorectal tumor biopsies. These come straight from the Cancer Institute of New Jersey, which we support all of the molecular diagnostics and molecular oncology work for the state of New Jersey. There are partner and development approaches like this, more specifically the personalized medicine initiative there. We ran quad sets of samples, 4 of them. We wanted to look certainly at tumor and normal adjacent and identifying variants, but also we wanted to look at fresh frozen versus FFPE. And as I mentioned this afternoon, you know, every day people say, well, I have some samples that are fresh, some that are frozen, some that are FFPE, so certainly we can analyze them all together, right? And then they want to know why, you know, their gene expression data might look differently, or, you know, why their sequencing data might be different. So this gave us an opportunity to see, you know, what utility is there for us to look across different sample types, you know, for this kind of analysis. We used the minimum sample input that we had experience with. We compared this to the LifeTech Ion AmpliSeq hotspot version 2 cancer panel, which we run a lot of. We certainly ran the RainDance ThunderBolts cancer panel, which was the reason for the evaluation, at 10 nanograms or less, and the Ion AmpliSeq cancer panel at 20 nanograms, only because that was really the comfort zone from the intensive evaluation that we've done over the last couple years now. We then use digital PCR validation for the detected variants. And down here is just a screenshot from some TCGA data in 2013. And again, I'm just going to focus on a few of the KRAS and P53 mutants, which again, because these are our blinded samples that we use for this evaluation, unlike some of the other talks that you're going to hear about, we don't have the clinical history to correlate, right? We have the bare bones, and it's nice to be able to have collections like this to do this. So just one more comment on workflow considerations, because that's important to us. On the left-hand side, you can see the ThunderBolts Workflow. Truly, it's a 2-day process with about 4 hours of hands-on time, which is pretty remarkable considering all of the steps and the additional QC that we do for some of the competing technologies. And the thing to really underscore on this slide is the time to technology implementation. So from the time of inception and our execution of the MOU with RainDance and looking at this technology pre-commercial launch and agreeing to evaluate it, we literally had sequencing data in 2 days. Even though we had someone from RainDance that walked us through this for the first time, the setup was no different than what we were doing for digital PCR on the source. And I can't say that about some of the technologies that we've run. By comparison, although in all fairness, it was when AmpliSeq was first getting started, it was just a little over 3 months to validation of AmpliSeq for the cancer panel before we could offer it on a commercial level within our lab services group. So that's a big deal for us. So most importantly, if we look at some of the performance data, the top table is the RainDance ThunderBolts results. The bottom is the Ion AmpliSeq from the hotspot version 2. We saw significantly increased reads across both. These are averages of fresh frozen tumor and normal adjacent and FFPE tumor and normal adjacent, which was great to see given the starting amount of material. In the beta version, which is not the commercial version, we saw 100% coverage up to 100x in all of the samples. So the averages obviously collapsed across the samples that were run, and even quite respectable data at 500x. The one thing that is striking is the uniformity, and this is really what we were looking at, knowing that our FFPE samples, will never be perfect. They're kind of beat up. Granted, some FFPE samples are better than others, but it was this, lack of variability or higher uniformity which gives us a lot of confidence moving forward compared to other targeted sequencing approaches we use. Now, the Ion AmpliSeq performs very nicely and as advertised, but for us, some of the workflow differences and this difference in uniformity is something that was really intriguing to us when we started to look more closely at the ThunderBolts data. But at the end of the day, it's really about the variants that we drill down and look at. And I thought that really the coolest thing that was part of this evaluation was looking at the correlation of variation frequency, not just from the sequencing data, but what we were able to validate with digital PCR on the raindrop. So if you look at a few of these samples, and again, you can see that AmpliSeq is not a poor performing technology. Really what we're looking to do is enhance on that performance by using ThunderBolts, which we were able to validate here, and really shorten our time to workflow, and put a technology in place that will allow us to put it in an enterprise level for all samples. So if we look on just the ones that are highlighted in red, you can see that the variation frequency, although for this KRAS mutant up on top for FFPE correlates nicely, you can see a much tighter correlation in variant frequency for in the fresh frozen tissue. Now, I do have one data point on the bottom, which actually shows AmpliSeq picking up a variant at a low frequency, and then actually validating that with the same DNA for digital PCR. And this was not detected in the ThunderBolts panel. So it turns out that because of the increased amount of sample that was needed to run AmpliSeq, we actually had to go back to that sample, cut more slices, extract DNA, and rerun it. So what happened was we got to the point in that sample that we actually were screening effectively a different part of that tumor sample or that biopsy, which are very small. And I just underscore that because this is like a real world scenario. When we have samples that come back that people that need more, we don't know if we're going to get the same complement, especially for something that's a very low variant frequency. If we had taken that sample and had enough to run everything from one extraction, I don't doubt that this wouldn't have validated the way that it did. But because it was from a separate sample, it, we saw different in results. And then this is some of the P53 data. Again, along the same lines, if you look at the data highlighted in red, we saw a very tight correlation and variant frequency when compared to digital PCR. And again, this was something that was, we didn't expect would be this good, but was a real positive outcome from the analysis. So if we look at our DNA QC analytical workflow, the top for the old there is really a manual workflow that a lot of smaller labs do. There's absolutely nothing wrong with it. What we do at the RUCDR is we automate this process through different functional and analytical quality controls, with the piece de resistance being a SNP panel that we've developed that looks at quality control for ethnicity, gender, sample performance, sample uniqueness. But it kind of ended there because we knew that people were using those samples for genotyping. But you know, what our workflow looks like under construction not only changes some of the chemistry now as we look at these analysis, using a different chemistry for looking at blood, fresh tissue, and FFPE samples, using the same automation infrastructure, but integrating now technologies like RainDance, which will allow us as a part of the DNA workflow to generate these panel results as a part of that process. So sample collection, accessioning, QC, storage, and providing this data that allows us then to have our investigators identify samples immediately that now they're going to go and look at on a much deeper level, all the while preserving that primary sample. So we don't have 50 labs doing the same screen, but allowing them to really target the samples they want, because it's our responsibility to preserve those primary samples. And again, using the same technology, being able to do target enrichment as well as digital PCR validation for those samples. So, you know, the need for biobanking's already defined. Biobanks are growing by leaps and bounds. The power for our research, the power for the development of technologies are the people that have the samples. And we work with a number of different federal and private programs so that we can get standardization across the community. However, for technology integration, that's going to be critical for molecular profiling on an enterprise scale. And what you will see is some of this profiling is being pushed onto that repository side of the framework, because that data is going to be critical in making those upfront sample assessments. So specificity and sensitivity is mission critical. You know, that is the first cut, which we were very happy with. Analysis and validation, so the integration of both of those applications is a huge advance for us. Efficiency and cost is what makes this operationalized, right? And like I said, there are a lot of technologies that we've looked at that have been very hard to bring in on enterprise scale, but this it should really be able to enable us. So the raindrop truly does enable, at least within the context of our program, the integration of sample QC by using digital PCR to make sure that we have performance and sample uniqueness, doing the targeted molecular profiling to create that data resource that goes with every sample, and then validating those targets all within a single platform using, you know, a various suite of RainDance technologies. So I see that everyone's been served dinner, right? So that's good timing. I just have to recognize certainly our contributors and collaborators from the Cancer Institute of New Jersey. For those of you who don't know, there's no longer a University of Medicine and Dentistry of New Jersey, which was by far too long to say anyway. It's now Rutgers Medical School, and Cancer Institute of New Jersey is now formally a part of our larger program. Bob DiPaolo is the director of CINJ. Sridhar Ganesan and Lorna Rodriguez head up the translational and personalized medicine initiatives. Jay Tishfield's the RUCDR CEO, and Mike and Wasi are two people in the lab that generated all of this data. So with that, happy to answer any questions. Sure.
In P53, for example, there's a lot of deletions, insertions that happen.
Yeah, so you know, we need to look at that more closely, but we need a larger sample set to do it. And you know, these samples, again, were chosen randomly without a lot of the information. We're now going to enter into a validation with a couple hundred samples that we have data on from the foundation panel we were running, although we're transitioning that more also to a homegrown larger panel. And that's exactly the first things that we're going to look at in P53 and a number of other targets in qualified samples. So this really was more to look at the efficiency, see what markers were identified and validated. But some of those things need to be addressed very specifically, and they'll also be validated using a deeper sequencing before we put this in play as a screening tool. Looking at the data quality, I have no reason to believe that's not going to be the case, but I don't have that data to show here.
So you have data showing sensitivity and specificity as a function of variance in frequency. So as you go from, say, 1% to 10% to 25%, well, how does that trade-off occur? What does the curve look like?
Yeah, again, we can only reflect on the data that we have here, and some of them were very high, and I think the lowest that we had was in the high 20s. There was one low that was the 5% that it showed up in the AmpliSeq, but validated by digital PCR. But digital PCR, we've looked at that, and the correlation is very tight based on the quality of the sample, if you will. What that's going to be against ThunderBolts, we don't know yet. And again, this is going to be part of this larger evaluation. If you look at the numbers, I would find it hard to believe that they're not going to continue to be tightly correlated. What I would have expected to see would have been a much lower result on the sequencing panel when compared to digital PCR. But they were extremely tight at 3 different levels. We had in the 80s, probably in the 40s and the high 20s or so. I think that might be a little more suspect when we get down into the single digits, but we have to run more samples to be able to determine it. But I have high hopes.
So you're doing spike-in controls and maybe half-half samples, spiked into different frequencies?
So we haven't begun any kind of clinical validation for this. And in our CLIA lab, to put this in play, because all of these samples will be run in the CLIA component for the RUCDR, we will do that. And that's typically what we do is spike-ins. We use a variety of hat-mat mixtures to be able to get at those frequencies, just looking at them, because you're never going to find them all naturally occurring. We haven't started that yet. I mean, this data is like a week old, and it's exciting. And these were from real clinical samples because we wanted to see what those comparisons would be just out of the box. But we will have all of that data available over the coming months as we begin to validate the panel for this intended use. And yeah, it'll predominantly be spike-ins and a variety of hat-mat mixtures. And the good thing about having close to 20 million samples is we've got a lot of real estate to work with. So where the hat-map, where we don't have, we can fill in for a lot of those.
Did you start from 10 nanogram as they advertised?
So, in some instances, we didn't even have 10 nanograms. So in the sample that we had run out of, basically, we ran it neat in ThunderBolts, which was sub 10 nanogram. We had to do multiple extractions for some of these to get enough for AmpliSeq and then be able to do the validation for those. So we ran 10 nanograms, right around 10 nanograms, some a little bit less, some a tiny bit more, but certainly not even approaching 15. And for the AmpliSeq, it was 20 nanograms and up.
So 10 nanograms is like 3,000 genomes, right? Or something like that.
About that.
So when you sequence at 10,000-fold, you start sequencing the same thing several times, correct?
Yep. Well, you can. So if you look at the data that led to those averages, I don't have it up here to show, but we actually accounted for the increased number of sequences. And when you look at the efficiency for sequencing, you also have to look at the relative amplicon size, right, as a function. So that was all factored into the normalization for those calculations. And what we did is, I think all the speakers have analyzed the data in a similar way so you can see it, comparably. But you're right. We could basically, if the barcodes existed, and I think that they do, is from what I was hearing in an earlier conversation tonight, is actually run more samples, bring those reads down to still achieve similar coverage for what we have here. It's a very hard direct comparison. In fact, these AmpliSeq samples were run on a proton and not even on a PGM because we wanted to boost the amount of reads that we got to make it more directly comparable to the MySeq given the fewer number of samples.
Yeah, Andy, as your initiative, if I understand, is to genotype and mutational profile of the biobank itself going forward, and I imagine one day going back through 20 million samples, which sounds like a Sisyphean task. But the question is, do you think that for something of that magnitude, and it's going to continue prospectively with a lot of samples coming in every week, is it more efficient for you to have tissue-specific reagents, or to go across the board with all 50 or whatever?
Yeah, that's a great question. And the way that we approached this in the past with some of our SNP panels, where it kind of served, that purpose in some respect, was that, 75% are locked down, and 25% we apply kind of a custom approach, if you will. So when we look at the RUID panel, which is that 96 SNP panel that we run on our biomarks, there is a certain percentage that we put in for markers that we might use for inclusion or exclusion criterias for specific trials. They might be tissue-specific, marker-specific. So I'll give you an example. We recently did a study with the Army STARS for post-traumatic stress disorder. These were blood samples. We collected 100,000 soldiers. After all of those samples were collected, we had to go back to 100,000 samples, run a few markers to decide which ones we would do deeper analysis on. That's not very efficient. It's not very cost-effective. You can try reasoning with the Army. I really hope nobody's here from the Army. I want to continue to get funding. But this is how people think, and these kinds of technologies enable some of that information to be evaluated up front. So what I envision is something that is, more comprehensive, but as you alluded to, having a percentage that might be specific for the study. So the vast majority of what we're screening will apply to every sample, but there is the possibility for specific trials or tissue types to make it more unique. And again, this is just a tool that will allow investigators to focus in on the samples that meet their criteria, not waste additional sample by doing more screens, and do a better job at identifying what their study participants will be, because there are more samples than anybody can afford to run.
So are you planning to run them on MySeq?
What's that?
Are you planning to run this on MySeq, just to have comparison between the...
So this was run on MySeq for this evaluation. However, we can't ignore employing some of our HiSeqs to do this just based on cost. So we don't have our next seeks yet. They're coming. That would be a function of cost. I don't think there's going to be a difference in sequence quality, but that will be the numbers of samples that if we can get it up to the point to barcode enough that we get the resolution we're looking for on the HiSeq, we'll certainly take the lower cost as a part of this routine screening program. However, I have no problem using the MySeq for those emergent samples that need to be run on much shorter order.
No, I was getting to the point that when you're comparing AmpliSeq was run on Iron Torrent versus or Proton versus the Illumina platform.
Right. No. So in this, the AmpliSeq was run on the Proton, and the and ThunderBolts was run on a MySeq. I'm sorry. I thought I might have mentioned that. We could have run it on the PGM, but again, we didn't want to cut off on the number of reads. We wanted it to be as directly comparable. So we kind of, I guess, stacked the deck a little bit in the favor of AmpliSeq because we have more experience with it. The data is what it is, and the uniformity was superior in ThunderBolts. Great, thank you very much.